So I'd like to welcome our next speakers, Dr. Paul Klenerman and Dr. Michael Fitzpatrick. Dr. Klenerman is our 2019 Established Investigator Award grantee and Sidney Truelove Professor of Gastroenterology at the University of Oxford. He is an immunologist who has extensive previous work in hepatitis C and HIV. Dr. Fitzpatrick is a clinical lecturer in gastroenterology and nutrition at the University of Oxford. His research interests are gut immunology and disease. And if you're looking for a new podcast, Dr. Fitzpatrick co-hosts a podcast on GI research called Gut Instinct, the GI Research Update. He said last night that there's only 3 episodes so far, but maybe he'll get some more ideas here. Dr. Klenerman and Dr. Fitzpatrick will talk more about their research into killer T cells that cause tissue damage in celiac disease. Welcome, both of them.
Great, thank you. And firstly, thank you to Beyond Celiac for inviting us and particularly for funding us. So it's a very exciting area to be in. And I was funded partly because I bringing some of the work that we've done in viral immunology into the celiac area. So I'm just going to talk a little bit about why you would want to do that. And then Fitz is going to talk a bit about what we've actually done so far. So, viruses are, as we have learned from the pandemic, they're much smarter than we are. First, I just get a few very basic things across about kind of viral immunology. So I work in a building called the Peter Medawar building, and this is a quote from Peter Medawar, who is also an immunologist who studied kind of tolerance. And he didn't have a very good view on viruses. The point about that from an immunological point of view is that the immune system, in order to fight viruses, has to set up tools to interrogate the interior of cells. And so viruses are essentially cleared through a slightly different mechanism for bacteria. But I mean, as we've all learned from the pandemic, viral infections have a huge impact on human societies. And Rolf Zinkernagel, who I work with in Zurich, wrote this famous article in Science about 20 years ago now, just sort of pointing out that you could learn almost any aspect of immunology by studying what viruses have to teach us. I think he's probably right, so hopefully this can be applied to celiac. So just to kind of run through how a virus grows in the cell, it has to enter and then replicate using the cell's machinery, essentially. And then it generates multiple copies from each cell. That's a picture of HIV exiting a cell. It connects the cell without causing any damage necessarily. So that's the kind of process that the immune system needs to interrupt. And to illustrate kind of the tension that the immune response is under in trying to facilitate this, particularly in the gut, I just take this illustration of a sort of, that you imagine your body's a sort of stately home in the UK. So in the back of this picture, you can, just in a very faint outline, you can see Oxford and the John Radcliffe, which is a sort of white building on the top. And this is not my house. It's or Fitz's, in fact. It's Blenheim Palace, where Winston Churchill was born. And so just imagine it's a kind of great big thing with grounds, essentially. And there's a party going on. So we're all taking in food the whole time. Guests are coming in, but the virus is not invited. So that's the issue. So the immune system has to be able to distinguish between things it's not bothered by from viruses which are going to cause a problem. So how's this sort of essentially managed? And most of the time it works really well, of course. So that's a kind of map of the situation. One point to make, and this is something we're really learning and Fitz will illustrate, is that the immune system isn't really a kind of extra sort of standalone system from the rest of the body. Every cell in the body is involved in an immune response, or any cell can potentially be involved. And a lot of the host defense includes barrier structures. such as the epithelium in the gut, which they've all got their own role to play. But the first line defense, when you get an intruder, so in other words, somebody that you don't really want invading, is the guard dog, and that's essentially your innate immune system. So that's got to be triggered in order to trigger any kind of fundamentally useful adaptive response. And the innate response is kind of like the guard dog, so they have a very good sense for the quality of an invading virus. So you can hear me, you can see me. The guard dogs have got very good senses for the presence of things that shouldn't be there. And then they can do two things. One is they can eliminate the virus directly, so that it doesn't get any further down the track. And the other is that they can call for support. So both of those are important. So essentially you want those dogs to be trained effectively to trigger in response to viruses, and you do not want them to respond to the guess. In other words, essentially food, gluten itself shouldn't require a kind of a guard dog response. So the things that we're looking at there are substances like interferons, which Fitz will discuss later, and dendritic cells, antigen-presenting cells, which trigger the very early events in the host response. So that's one aspect. And then once the process has got going, you've got kind of a fairly standardized type of activity called the adaptive immune response, which is specific for individual components of individual gate crashes. So these are a wide variety of cells, and as Fitz will illustrate in the discussion, we're getting better at kind of pulling these apart. There's sort of broad categories which we've known about for decades, and then the fine resolution of this is becoming a bit clearer with the new methods we got. But fundamentally, there are three groups. They're the killer cells, which has been the kind of focus of this work, and I'll give you an illustration of that. Some helper cells, which are the sort of logistics experts which get everything together, which are crucial in celiac. You're not going to get any response if you don't use those. And they're antibody-creating cells which mop up antigen protein that's outside the cells. So back to our analogy, the killer cells are going room to room. So they're looking at the interior of every cell, exploring it, asking, is this a safe cell or not a safe cell? And if it's not safe, they'll either kill it or they'll try and eliminate the virus without killing it. And the antibodies are kind of got a much more general role in eliminating circulating virus. So the killer T cells play a very important role in any immune response. And this is how they do it. So that if it's a virus, and it will come in and it's presented through a processing pathway on specific HLA molecules, so highly evolved antigen-presenting molecules to the killer cells. And then the killer cell will respond by triggering very sensitively and have a tendency to kill the cell. It can do other things as well. And the idea is that it eliminates the process of viral spread. So that's a problem. So you've got to really tune that really carefully. And the hypothesis in celiac is that this is detuned, and that's part of the issue. So this is a killer T cell. It's actually one of mine, which was grown in the lab. And the target is, in this case, a cancer cell. But it could be an epithelial cell in the gut if it was appropriately kind of targeted. And you can see there's a very tight association between the two cells. It's called a synapse. The little red dots are proteins which are able to kill. And then the other cell undergoes programmed cell death. So it's actually quite a controlled process. It's not sort of shooting up the cells sort of randomly. It's tightly controlled. And we don't really know enough about this in celiac disease. That was our feeling. Well, there's some ideas about it, but the real details are not really known. So I think the idea is that once a cell, a program of T cells has been generated, essentially a group of highly trained T cells, they'll go out into the periphery. If there's any problem, they'll address that very specifically. So you need enough. It's a sort of numbers game. Build them up, and then eliminate the problem in the case of a virus. And then you're left with memory. And the memory is very long term. It's actually not just in the sort of security room, it's out in the tissues, as we'll show you in a minute. And that is a sort of natural process for generating an immune response to anything. So, yeah, and we all have, and as you've been vaccinated against COVID, we got, a series of these for COVID. The vaccines work very well. And just as much as they make antibodies, you'll make T-cell memory. So that's the sort of outline, and that's the kind of led to the hypothesis. Now we spent years trying to figure out all the details of this. Does this tell us anything about celiac disease, and particularly around about the killer cells? And all the tools that we've used to understand how we generate these against different viruses, how we make vaccines, can very well now be applied to celiac disease and really push our understanding forward quickly. So over to Fitz.
I'm the second half of the double act, and I'm going to spend the next 20 minutes talking about the work that we have done and try and apply, I guess, our thoughts about how understanding viral responses in the body can inform our understanding of celiac disease and other GI conditions. I'm a gastroenterologist in Oxford, and I did my doctoral research in Paul's lab, and then have continued on with this project more recently. So thank you for Beyond Celiac for your support for this work. So I'm going to briefly run through our current model of celiac disease immunology for sort of a general audience, and then I'm going to discuss the kind of key questions that we had about these killer T cells and what additional things we can learn about them, and describe hopefully the methods that we used in a nice clear way. And then I'm going to talk about two things in particular from the research. One about the cells that line the gut, the epithelial cells, and then the CD8 killer T cells that we're particularly interested in. And there's a number of bad celiac-related puns throughout the presentation for you to look out for. So here is my sort of general explainer for celiac disease biology. We've got the epithelial layer, the lining of the gut, and gluten that you ingest is broken down by enzymes into peptides, short protein fragments, and they get sort of taken in through the epithelial layer and then altered by this enzyme tissue transglutaminase 2 that some of you will have heard of, and it's these deamidated gliadin peptides that are taken up by antigen-presenting cells, this innate part of the immune system, and they get presented in a manner very similar to a viral particle, and they get presented to what is undoubtedly the MVP of celiac immunology, which is the gluten-specific CD4 T cell, of which there's been a huge amount of research over the last 25 or 30 years, and I think we know more about this than really any other disease-specific T cell population in really any autoimmune condition. And then something happens. Something triggers this cell to take on a pro-inflammatory phenotype, rather than, as normally happens, to become tolerogenic and tolerate food antigens. And we think that that's a viral infection. We think this is a viral trigger that shifts the immune system into this kind of pro-inflammatory response, where a whole load of cytokines, chemical messengers, are released, and they drive the cells that we're particularly interested in, the CD8 killer T cells, many of which are in the lining of the gut, called intraepithelial lymphocytes. And it's them that directly cause cell death in the lining of the gut. One of the things in this model is that we think, or what's been described, is that these CD8 killer T cells are killing epithelial cells via a very kind of a blunt instrument, a couple of what are called natural killer receptors that recognize stressed cells and then kill them. It's a sort of a kind of a blunt tool. And in this picture, the CD8 killer T cells are kind of dumb foot soldiers under the command of the gluten-specific T cell. And they're using very sort of primitive weapons to just respond to stressed cells. But what we know from viral infections is that there is this other part of the CD8 response, which is much more like a sort of an elite special force, where they've got highly targeted tools, these T-cell receptors, and they can recognize really specific antigens. And we see this in viral infections, but it's not really understood how that part plays a role in celiac disease. And so that's what we've been particularly interested in. So some of the questions we've had from this in this research project is to understand what those CD8 killer T cells are doing in celiac disease, what kind of CD8 T cell there are, because they come in a few different flavors, and how are they using this highly targeted tool, this receptor, the T cell receptor, in celiac disease. And one of the main tools that we've used, and that's the main thing I'm going to talk about today, is something called single-cell RNA sequencing, which is not a terribly intuitive method. I'm going to take a couple of minutes to describe what we do, just so that everyone's kind of on the same page. So RNA sequencing has been around for 20, 25 years, and is looking at what genes are activated in cells or tissues in a particular condition. And what we do is we take a piece of tissue, maybe a duodenal biopsy, and that's made up of lots of different cell types. Here, 3 cell types are shown, and each one is making, is using particular genes and turning them into proteins. And in bulk RNA sequencing, we take the whole biopsy, we put it in a metaphorical blender, and then we extract out all the RNA. And then we just count them, and we say, well, there's more of this red RNA than there is this black RNA in this particular sample. But we can't say what cell is making which RNA molecule, which genes are being expressed in which cells. So in single-cell RNA sequencing, we take the same sample, but we effectively put each cell into its own little reaction chamber, and we do that same thing. And so now we can come up with a matrix where we can say which cells, which very specific cells, are making which genes in what condition. So we can really unpick some of the interactions that are going on in disease. And this is the project that we've been working on for the last few years, which is a large cohort study of single-cell RNA sequencing in celiac disease. And we've studied 2 cohorts, which I'm not going to discuss in great detail, but an adult and a pediatric cohort using 2 different single-cell RNA sequencing technologies. And we've been looking both at duodenal biopsies, but also blood samples, and particularly in the adult cohort, we've been looking at both the epithelial cells, the cells that line the gut, as well as the immune populations. So I'm going to just present a small amount of the research focusing on those epithelial cells and the CD8 cells and what we've learned from that. So the first thing is that epithelial cells, lots of cell types, are slightly like ice cream, in that they come in lots of different flavors. You know, you go for ice cream, the first question is what flavor you're going to have. And we can see that in epithelial cells lining the gut. There's a lot of heterogeneity there, and this is a plot, this is a 2-dimensional representation of the thousands of genes each of these cells is making. Each dot is a cell, and we can identify all the different subtypes of epithelial cells and the genes that they are making, the genes that they're expressing in those cells. And so there are the main cells, the enterocytes, that are the absorbative cells in the gut, pictured in green. There are the precursor cells, so the stem cells, and then ones that we're interested in called transit amplifying cells, which are precursors to those enterocytes. And then there's lots of really interesting secretory populations in the gut, including ones that make lots of gut hormones that modulate metabolism. And we can look at what genes those cell populations are expressing and what they're doing in different disease states. And one of the first things we saw is there are really different proportions of these epithelial cells in health and disease. And so in this middle plot here, the same kind of 2D map of these cells, the larger light-colored dots are areas that are overrepresented in active celiac disease. And interestingly, it's particularly these transit amplifying cells, these precursor cells that are on the way to developing into absorbative enterocytes that are increased. And that's one of the explanations why there's a malabsorption aspect in celiac disease, because these cells have not yet developed all the kind of cellular machinery needed for proper absorbative function in the gut. Not only are these populations altered in proportion, but they're doing different things. So in celiac disease, nearly double the proportion of the epithelium is actively proliferating. So this is a sort of hyperproliferative sort of cycling state in celiac disease as they're desperately trying to replace the damaged enterocytes. They're also expressing different sets of genes, and what's really interesting is that a lot of these are what are called interferon-stimulated genes, so gene sets that respond to these chemical signals that we generally associate with viral infection. So these interferons, these cell signals associated with viral infection, are playing a role in the epithelial responses in celiac disease. And they're also expressing a lot of these HLA molecules, which present antigens to different cells. So at the sort of halfway point, the epithelial cells, the gut lining, is really radically altered in celiac disease, and they're shifted to this less well differentiated state. They're busily dividing, and they're expressing different sets of genes, and we can start to understand what they're doing in response to celiac disease. So the second population I wanted to talk about is all of the flavors of CD8 T cells. So these are the killer cells that we've talked about earlier, that we find in the lining of the gut and also the deeper levels in the gut. And we can identify a number of different states of these cells, different types of them. And what's really interesting in the gut is that a lot of these cells are what are called tissue-resident memory cells. So these are two markers associated with tissue residency that I'll discuss in a sec. One of them at the top is called CD103, which is one of the key markers for these tissue-resident populations. Tissue residency is a really interesting concept, which is probably best defined by the Eagles. But by the line, you can check out any time you like, but you can never leave. So these CD8 T cells are memory populations in the gut. They're long-lived, they're sitting there, but they've lost their hotel key card. They can't escape. So they're sitting there as kind of sentinels of the immune system in the lining of the gut. And it's these cells that seem to be playing a really key role in celiac disease, because particularly this green population at the bottom, this tissue-resident memory type 2 that we've identified, is radically increased in proportion, both in active and in treated celiac disease, in both the adult and pediatric populations we've looked at. There are also a lot of CD8 cells that are cycling, that are proliferating, and those cycling cells look like they've come from these tissue-resident memory cell population. And those, something slightly strange has happened, those cells are producing are expressing different genes than in both active and treated celiac disease and in healthy controls compared to active celiac disease. So genes associated with activation and inflammation, and that gives us insights into potential pathways that could be targeted for therapeutic strategies. What we can then start to do is to start to try and tie together who's speaking to who in the gut. Because that's the great thing about these data sets, is we can start to say, well, if you've got the receptor on this cell type, and you've got the ligand in another cell type, it looks like these two populations are interacting and talking together. And so, for instance, one of the things that we've kind of delineated is this particular receptor that's well described as a gut homing receptor, CCR9, is highly expressed on some of these tissue-resident populations, and its ligand is being produced by these precursor cells that are increased in celiac disease. And what we're finally doing at the moment is trying to integrate that with kind of spatial data, so trying to work out where these interactions are happening. So just from the last few weeks, we've been using a method called spatial transcriptomics, where we take, this is a single gut biopsy, about 3 millimeters across, and what we can do is we can stain it for different cell populations, and then we can zoom in on particular structures, like crypt structures, and then we can try and work out what genes are being expressed by particular cell types in a particular place. So hopefully what we can do is find out what's happening to different cell populations in terms of the genes they express and their proportions, how they might be interacting, and where they might be interacting. And I think it's those interactions which are really going to bear fruit in our understanding of celiac disease, immunology, and in particularly the CD8 T cell response. So I hope we've convinced you that understanding viral biology, how the body responds to viruses, can inform our understanding of immune-mediated disease like celiac disease, and shared some of the insights about the CD8 and epithelial populations that we've had from this grant. So there's, this is very much team science, and we've got an amazing group of collaborators in Oxford. I want to particularly thank collaborators in Paul's group and Agne and Tana Vicute, who's a talented bioinformatician who we collaborate with as well. And then particularly to our funders beyond celiac disease, without whom this wouldn't be possible. So thank you all for your time. I don't think I overrun, which was a surprise.
So I have maybe two questions in a way related to your work. First of all, I think your work clearly illustrates how important is cross-disciplinary research, right? How much you can learn from viruses or another discipline applied to celiac. And also, I think point out to the fact that certain assumptions that were made, for example, about celiac, all this work about CD4, it doesn't tell all the story. So specifically looking about your finding on the CD8 elite subpopulation and so on, I was wondering if you can, again, express some view about how you think this can be applied both into development of treatment or even kind of biomarkers for diagnosis of disease. And the second question that I have is, it's fascinating, this link, of course, with virus and what happened. We know certain virus triggers celiac and so on. And there is a lot of interest by us and others, I'm sure your group as well, about the gut-brain axis, connection. Do you think that these changes that you see in the mucosal barrier and the TCD8 can also have some effect on the brain in patients with celiac disease?
Thank you. So your first question, I think, I guess our hypothesis from some of these data is that these tissue-resident cells, I haven't included all of our findings in this presentation, but it does look like they are using this sort of highly specialized receptor, this T-cell receptor. There's signs that that pathway is activated, and that the types of T-cell receptor, the T-cell receptor repertoire of those populations is quite shifted, which suggests that they're responding in an antigen-specific way. And so while the previous methods of using NK receptors are clearly valid and true, I don't think they are the complete picture. And then that raises really interesting ideas of, well, what antigens are they responding to? And we're trying to investigate that at the moment and what they might be presented by. And that's a really tricky thing to unpick. But actually, it's really important for trying to work out whether therapeutics will work and how they will work. Because if we're just targeting, for instance, the CD4 response or the presentation on DQ2, let's say, to the CD4s, if that pathway isn't actually the complete picture, then it may be that just targeting that pathway will only have a muted response. And it may be that there are other pathways that need to be targeted. So I think that's the main thing from the translational point of view. The other point is, and I don't want to rant too much about it, but I'm really fascinated by ideas about tissue residency. And these cells that are stuck in the gut, and CD4s are often stuck in the gut as well, are different from the ones we see in the blood. They respond differently, and some of the treatments and things that we do may look different in their responses in blood and gut, and we just don't understand enough about how these cells in the gut work. And so I think that's a really interesting thing, that we need to understand more about these resident cells. In terms of gut-brain axis, I'm looking forward to Ian's talk later, but I think it's really, really fascinating, and there's been research about things like Parkinson's and the link between the gut and the brain in things like Parkinson's disease, and what might mediate these things, and the wide breadth of neurological symptoms. I guess one thing that interests me is the symptom that is most common in my clinical practice is brain fog, And is that, what is the basis of that? And could it be related to, for instance, inflammation or cytokines or chemokines released from particular cells in the gut? So yeah, I think it's a really interesting area, but I don't think I've got any answers for you, I'm afraid.
Yeah, very nice presentation. Can you tell us a little bit about what you saw in the CD4 compartment? You know, you focused on the epithelial cells and CD8, but are CD4 cells undergoing activation at the same time in the gut tissue, or do you think
Yeah,
that happens?
I can give you a brief rundown through the CD4 data. So, as has been previously reported, one of the interesting things is there's an increase in regulatory T cells in the gut, and that's been shown before, but they're clearly not doing a terribly good job. And that's really interesting. So there's increased numbers of regulatory T cells, but they don't seem to be responding appropriately.
It probably depends on the ratio of regulatory T cells to effector cells.
And then there's a population, and I think it expands on, our data expand on some of the work that's been done from Mark Davis's lab and Ludwig Solid's, that there is a population that have a really distinct phenotype that doesn't actually fit in with a typical sort of Th1 cell, that has markers often associated with follicular T cells or peripheral helper cells, which is likely where the gluten-specific T cells lie. And those cells are clearly expressing molecules that underpin their interaction with B cells and plasma cells. And so, yeah, we've described this really interesting population, and I think it builds on some of the work from those labs. So, yeah, we're really interested in those data as well.
Thank you both so much.
See?